when everything else doesn't work, colonics, you sometimes have to reset the bowel, and because there can be a lot of impaction of fecal matter over years of build-up, and so then there's no surface for the beneficial bacteria to really thrive and live in. And so with that just involves colonics and flushing out the bowel, so you've got a nice clean surface to work with and get the bowel working really well. For a lot of our chronic patients, we often don't see a breakthrough in their health condition until they've done a series of colonics, and we refer them out for that, and then we'll often see a breakthrough in their condition and an improvement in blood parameters.
We followed on by some important work by one of our collaborators, a gastroenterologist in Australia. He had treated 9 children with autism who had chronic GI problems, and he treated them with microbiota transplant. Unlike with C. diff, where one dose, one time, 90% cure rate, he found kids with autism were much harder to treat. So he treated them every day for 3 months and found that he was able to slowly, gradually improve their GI symptoms, and surprisingly, also found improvements in their autism symptoms. So with the help of Dr. Brody, we designed a clinical trial, phase 1 FDA-approved clinical trial, in which we treated 18 children with autism, and all of whom had chronic GI issues, chronic constipation, chronic diarrhea, or alternating between the two. We treated them first with a special antibiotic to kill off pathogenic bacteria, then a bowel cleanse like you'd have with a colonoscopy to remove any remaining bacteria. Then we treated them with microbiota transplant from healthy, carefully screened donors. We did that every day for 7 to 8 weeks. Slowly, gradually, the GI symptoms improved. 90% of our children had great improvements in GI symptoms. But surprisingly, there was also a slow, steady improvement in autism symptoms, about 25%. So we were really pleased with that. It's an open label study, so there's some placebo effect. So we were pleased with those results. We followed up 8 weeks later. We saw those results were still stable. But then I thought it was done. Great. But then a year later, a family came up to me and said, Professor Adams, my son's doing better than ever. Then another family came up and said the same thing. And a third family said the same thing. So I said, okay, we need to do a follow-up study. So 2 years after treatment stopped, we followed up with every one of the 18 participants in our study. None of them had dropped out. And what we found is that most of the GI benefits had continued. There was still about a 59% improvement compared to the start of the study. Wow. What was really interesting is almost all of the families said there was just a slow, steady improvement in autism symptoms. The children were just learning a little bit more each day, beginning to catch up to their peers. So at the end of 2 years, our professional evaluator evaluated the families and said there's about a 47% decrease in autism symptoms. Most of the children started the study with severe autism. At the end of the study, most of them had either mild to moderate autism or even below the cutoff for autism. They still had symptoms. They weren't completely cured, but they had made big improvements. So it's just very exciting to see.
Well, I like to think about the lifestyles in addition to nutrition that could help our biome. And believe it or not, our gut bugs have a circadian rhythm. They have a night and day rhythm just like we do. And there's scientists that are doing research on the circadian rhythms of bacteria. And so when you don't sleep, your bugs don't sleep. And they need sleep. So it's really important to have that kind of lifestyle where you get sleep. And believe it or not, exercise also stimulates a healthy balance of the microbiome. You begin to see a trend here, don't you? So then the one that I thought was just scary, horrible, is that our use in antibiotics in America, 70% of all the antibiotics that are used in America are used to feed the animals from which we get our eggs and milk and meat and so on. 70% of all, and why do they do that? Because you give antibiotics to an animal and it gets fat faster. and it goes to market quicker. So 70%, ever wonder why we're getting more bacteria resistance? And think of all of you, you know, the first time you get a sniffle, you know, I got a cold, and, oh, I got to get rid of this. Oh, I better call my doctor and ask for a Z-Pack or something, you know, quick, a Z-Pack. Every time you treat your viral infections with an antibiotic, as in bacterial, it changes your biome. So if you do need antibiotics, be sure and follow up with your probiotics, your kefir, your sauerkraut, your kimchi, whatever you prefer. You've got to repopulate your bowel if indeed you have to use antibiotics.
This slide just illustrates the impact of two antibiotics to treat Clostridium difficile, which is a severe infection that is often acquired in hospital. So as a result of the outgrowth of this bacteria, we get sick, so we're administered vancomycin or metronidazole. And what you can see is the diversity, the number of bacteria we have in our gut is drastically reduced because the antibiotics have killed them off. But it's also killed off Clostridium difficile, which is a good thing, but you know, there's a consequence to this in that we wiped out a lot of our good bacteria. So too many antibiotics for too long have a very profound and can be a long-lived effect on our microbiota. We really shouldn't be too surprised by this because we now know that the gut actually contains an awful lot of the neurons, neural circuitry that's present in the brain. And it's often thought to be the second brain. I mean, it has a very large number of neurons, 500 million, and it produces lots of neurotransmitters. And, you know, here's some sort of evidence that sort of links the gut to the brain. So brain-dead people, their stomach functions normally for quite a while. It's almost an inherent activity. Anybody that's taken major pain-killing drugs, like morphine, for example, you know, there's a risk of constipation. It shuts down motility in your gut. And emotions and feeling are intimately associated with bowel function. Right? We've all had the butterflies in the stomach, you know, that nervous gut reaction. Well, that's all those neurons in your gut that are firing away. And when you look at the structure of the nerves in the gut that make up the enteric nervous system, you know, these are the neurons or the dendrites here in silver, the white color. This is our gut tissue, and you can see that when we superimpose these two, the nerve fibers actually penetrate and intermix between all our gut tissue, and they actually look like they're actually protruding into the lumen to be able to sense perhaps the presence of factors that they can respond to that are in the gut lumen that could be made by gut microbes. And the vagus nerve is ultimately the route by which all this signaling in the gut leads into the brain. So all these signals here that the enteric nervous system responds to are fed into the brain via the vagus nerve. And so we know that if the vagus nerve is blocked or damaged through injury, profound effects on appetite and eating. In fact, it causes drastic weight loss. So it's clearly a regulator of body weight. And vagus nerve stimulation by hormones and neurotransmitters in the gut can drive excessive eating behavior. So overstimulation is not necessarily a good thing. And not surprisingly, gut microbes can actually regulate how much of these neurotransmitters and hormones are produced in the gut. And they can manipulate this to their own advantage by producing things that can block or stimulate the nerves in the gut. So microbes control eating behavior by influence signals that are delivered to the brain and by the vagus nerve. And two of the most important ones are dopamine and serotonin. So dopamine, associated with rewards, pleasure, compulsions. Serotonin regulates our mood, our memory, sleep, cognition. Dopamine, about half of the amount of dopamine produced in the body is produced in the gut. And some gut microbes can produce vast amounts of dopamine. And some of you may know that L-dopamine is used to treat Parkinson's disease. Serotonin is even more striking because virtually all the serotonin in the body is made in the gut. And gut microbes produce factors that can mimic or block serotonin action in the gut. And deficiency of serotonin is linked to depression. So I hope you can see that microbes, by manipulating just these two neurotransmitters, can profoundly influence our mood, behavior, whether we're anxious, whether we're relaxed, how much we sleep, how much we eat. And so linking this to a disease, this is interesting, this is a more recent study now, linking gut microbes to a disease that's called autism spectrum disorder, so autism. So we know from looking at patients' microbiotas that they're disturbed. They have alterations in the makeup of microbes, and also there are altered levels of what the microbes produce. And there's a mouse here that can develop autism-like syndrome. In particular, excessive grooming and vocalization is affected as it is in autistic children. And what this group that worked with this mouse showed that they could restore or treat this mouse by using probiotics, so live bacteria. And so the live bacteria altered the gut composition of the microbes, and it looked now more like normal animals. And this was linked to resealing of the gut. So these animals had a leaky gut, and it was the leakiness allowing these microbe-derived byproducts to get into the bloodstream and into the brain. But as soon as the barrier was improved, the leakage stopped, and it restored the normal levels that you would find in the serum, and it stopped or halted some of the features of autism.
As we begin to look at the actual genetics, this was the breakthrough that allowed us to actually understand our microbiome, particularly in our GI tract, because when I was just looking at them through a microscope, it was very limiting. You could only see just certain kinds. But now that we could actually genetically look at this. This all started about 10 years ago, and many of you are familiar with this, when they started looking at the genetics of the human. What are the genes? How many genes? What are the genes that we have? Through incredible research, it became apparent that we actually had like 26,000 genes. And I thought, wow, that's really cool. And everybody was studying the genes, and this was really wonderful, until they started studying the genes of a rice plant. The rice plant had 46,000 genes. What's that? You know, I mean, we're only 26, and they're 46? And so this was very humbling to say that the rice plant was more sophisticated than humans. So then about 5 years ago, everybody got busy, and they started to do the genome of the bacteria that resides inside of our body. Guess how many genes there are there? 100,000 genes. And so we begin to look at it. What are these bugs? Who are these? What are these? So there's like 100 trillion of them. When we think of cells of our body, our biome describes 90% of all the cells. We're only 10%. So we just heard how wonderful it is to look at the astronomy and be humbled. Well, I would suggest we probably just need to look inside of us and really get humble, because the other is way beyond this. So these genes are incredible. There's about 1,000 different varieties, and then when we look at the species and sort of like that, it's incredible how diverse this whole environment, this whole biome is, and it just resides in some just humble you and me. Well, okay, let's team up with our bugs. So we're 26,000 and they're 100,000. Wow. Now we can trump the rice plant. So we better stay joined with them. So we begin to do what actually does this biome do for us? So we first think of fermentation. Talk about a microbrewery. You know, right here in our right colon, right here, these bugs are actually fermenting, because this does a lot of good things, and they produce about the equivalent of a can of beer every day. And Oh, yeah, that's true. And so, of course, we handle it quite nicely and so on. We don't get tipsy with that much. And so in this process of fermentation, some very important things are created, and they're called short-chain fatty acids. These short-chain fatty acids are critical to our immune system. So if you breed a little mouse that has no biome, this little creature is very vulnerable to infections and so on. And so in many ways, this is quite dramatic. And we wonder, where does this biome come from? Because the little human, when he's just inside the uterus, he doesn't have a lot of bugs. This is kind of coming in question here, like right now. But so far, we've always thought of it being kind of sterile inside there. But when this little child goes through the birth canal and is breastfed, that is where the microbiome starts. This is critical to the life of this child. And our C-section babies and our babies that don't get breastfed, this is very difficult. So now, in modern places and hospitals that understand this, if the little child has to be born by C-section, a vaginal swab is actually taken and placed in the child's face and mouth so that it can actually become a microbiome positive creature. Otherwise, the immune system would not develop. So this is very, very vital. So that's kind of the positive side of the fermentation. But there's a negative side to this, and this is where, remember all these cells, these thousands of different kinds of bacteria. Like most of life, it's about a balance. And when they are not balanced, we call it a dysbiosis. When that begins to happen, we find that it starts causing diseases. You know the word irritable bowel syndrome. Some of you may actually have it. You know, diarrhea, constipation, abdominal cramps, all these kind of things. You know about colitis. You've heard of people that have colitis. These kinds of things are when there is a dysbiosis. There's an imbalance between the good bugs and the bad bugs. Also, we're beginning to understand that a lot of what is called autoimmune diseases, such as rheumatoid arthritis, multiple sclerosis, these kind of things may indeed be associated with an imbalance in our microbiome. So you begin to see how this science is beginning to progress. And a third area that I think is very critical is what we call the gut-brain axis. And when you look at animal studies, you can take, again, these little germ-free mice, and when they're born, they're kind of autistic. You know, they don't kind of hang out with their puppy brothers and sisters, and they don't eat well, and they don't do things well, and they're kind of autistic. But if you then transplant normal mouse biome into these little guys, they become normal. They just kind of hang out with each other and so on. You can see where this is going, can't you? And sure enough, it's already happening. So in Europe, there's some beautiful studies that are being done on humans. Here in America, there's some people that on the side are just giving probiotics. You know, probiotics have the microbiome in them, probiotic enemas to autistic children, and actually seeing some development. I have not seen good randomized controlled trials, but where there's a little smoke, there definitely can be some fire. Now, in the human studies, one of the things that we want to know, so we already know that the gut affects the brain. What we also want to know, does the brain affect the gut? And sure enough, when we find people that are under high stress, of course, nobody here in the audience, I'm sure, that was high stress. You know, that must be someplace else. But under high stress, the biome actually changes. And what we find is that there's a breakdown. So inside the gut, there's a nice little layer of mucus. So you have all this bacteria, and here you have your gut wall. And then this first layer of mucus, there is no bacteria. It's a barrier. The next layer is another layer of mucus that does have bacteria in it, and under stress, those mucus layers begin to break down, and antigens from the bacteria actually penetrate into the muscle wall and therefore into our circulation. So this barrier is broken down. And there's a great deal of study that's going on now to even look at microbiome and Alzheimer's. Alzheimer's is now being called diabetes type 3 because we know that those high insulin levels, those high sugar levels, all these things may well be contributing. So at first I thought, well, this is a little fringy. But when I begin to see that we have neuroscientists that have linked together, 30 neuroscientists in the Scientific American just published an editorial saying we need to take another look at Alzheimer's disease in relationship to the biome.